We will spend the next few minutes discussing some basic features of viruses, which of course are important for a variety of reasons. We'll focus on basic virus structure. We'll discuss some reproductive cycles of viruses. And in particular, we'll spend some time talking about the reproductive cycle of HIV. And finally, we'll wrap up this discussion with prions, which are an even simpler form of infectious material than our viruses. So let's begin addressing some very basic questions. So for example, what exactly are viruses? Are they considered alive or not? And how big are they? If you take a look at the image in the lower right, what you see are a few different things. First of all, this large red structure, that's a red blood cell. So red blood cells, they're fairly small to begin with, but then for comparison, in blue you see a few bacterial cells, which of course are even smaller than red blood cells, and even smaller than that are these small red dots. Those are viruses, just to give you a sense of how big viruses are. So viruses are extremely small, much smaller than most bacteria. Basic viral structure is illustrated in these images here. So if we begin with the image on the upper left, what we see is a so-called non-enveloped virus. Essentially, a virus fundamentally contains at least two components. It consists of some proteins as well as some genetic material. That could be DNA or it could be RNA, as we'll see a bit later. But again, viruses have to at least have those two components, proteins and some genetic material. Some viruses, slightly more complex, illustrated like we see on the upper right panel here. This is a so-called enveloped virus. HIV is a classic example. What this is again, it's just a virus consisting of proteins. We see these green capsid proteins here, some genetic material interior to that, but then surrounding the capsid is this envelope. It's basically a membrane, and essentially this is a membrane that's derived from a living host cell. So the virus doesn't produce this membrane, it essentially captures it from a host cell as the virus exits a living host cell that it had previously infected. Some characteristics of viruses are illustrated in this table. So if you consider organisms, they always, at least all the ones that we know of so far, they always use DNA as their genetic material. Viruses, however, can use DNA or RNA. Organisms, living cells, are defined by the presence of a plasma membrane. Viruses, however, do not have a membrane, again, unless they capture one from some other living host cell. Organisms can, of course, undergo gene expression. So transcription, translation can occur in organisms. Viruses, however, they cannot do that. The only way they can undergo transcription or translation is if they rely on some living host cell. So viruses have virtually no metabolic capabilities on their own. Again, they always have to rely on some living host cell to undergo all the metabolic capabilities required for reproduction of viruses. So as you can see, there are lots of features that sort of separate viruses from other things that we consider to be living organisms. Let's take a look at these viruses that are specialized for infecting bacteria. So-called bacteriophages, or simply phages. Again, these are viruses that infect bacteria. Let's use these to illustrate some basic principles of the reproductive features of viruses. So what we're going to look at as an example of these bacteriophage reproductive cycles are the so-called lytic and lysogenic cycles. So in this initial panel, what we see is the lytic cycle of a phage particle. So what we're looking at in yellow, this is a bacterial cell. Could be E. coli, for example. Bacteria, they have their chromosome, illustrated in this gray squiggly line. And what we see here in green is a bacteriophage infecting this bacterium. So essentially the bacteriophage, again it's a virus, injects its genetic material, for example DNA, into the living host cell. That DNA, once inside the living host cell, can replicate, and the genes on the DNA can be expressed. So transcription can occur, the mRNA is then translated to produce all the proteins, the capsid proteins and so forth, that represent part of the viral particles. Those proteins and copies of genetic material are then assembled into new viral particles, so new bacteriophages are produced again inside the living host cell. Eventually, those new viral particles are released from the host cell. That results in the direct death of this living host cell. But then these new bacteriophages are then released and from their perspective, hopefully go on to infect other host cells so that they can then reproduce even farther. So again, that represents the so-called lytic cycle of bacteriophages. A slight variant on that is a so-called lysogenic cycle, illustrated toward the bottom. Initially, the same things take place with bacteriophage injecting its DNA into a living host cell, but notice the bacterial chromosome incorporates the viral DNA directly. So the viral DNA gets incorporated into the host cell chromosome, and instead of those viral genes being expressed right away, they essentially stay latent. Those genes aren't expressed right away, but notice then that every time, and again, it doesn't kill the living host cell directly, so that every time that bacterial cell divides, it results in a replication of the viral genome. Then at some later point, these cells can be triggered to undergo the lytic cycle, like we see illustrated toward the top, which again would then result in the formation of new phage particles that would be released and again would eventually end up killing the host cells. But again, the lysogenic cycle, illustrated toward the bottom, does not result directly in the death of the living host cell. We already mentioned previously that the genetic material in viruses can either be DNA or RNA. As a classic example of an RNA virus, let's take a look at HIV. HIV is considered one of these retroviruses. And what does that refer to? Well, that refers to viruses whose genome is RNA, but then have enzymes, specifically reverse transcriptase, that can take the viral RNA and produce a corresponding piece of DNA. So they essentially are sort of going backward compared to the normal flow of information, which is typically DNA to RNA. So again, retroviruses can take the viral RNA and produce a corresponding DNA with this enzyme reverse transcriptase. So let's take a look at the infectious cycle of HIV. To begin with, let's just take a look at the structure of HIV illustrated here. So HIV is an example of an enveloped virus. So here we see this outer envelope, which again essentially represents the cell membrane from some former living host cell. On the outer surface, there are these glycoproteins. I just want to specifically point out GP120, glycoprotein 120. We'll see that plays a very important role in the reproductive cycle of HIV. Interior to the envelope, we have our capsid proteins illustrated in green here. Interior to the capsid, we have viral genome, which again in this case is RNA, illustrated by these yellow lines. There also might be a few enzymes present within the capsid. So for example, here we have these green dots. Those represent reverse transcriptase. Also another enzyme I'll mention is integrase, simply illustrated graphically here by this blue box. So that's the basic structure of HIV. Let's now take a look and see what happens when HIV infects a living host cell. Keep in mind that in humans, HIV tends to infect various types of white blood cells, for example, T cells. So what we might be looking at here is let's say, a large T cell. T cells play a very important role in the immune system of humans. So let's begin in the upper left here, and notice here's an HIV particle. Notice that the GP120 on the outer surface of HIV is recognizing and interacting with this structure in yellow. This is so-called CD4 receptor. So this recognition between this receptor, CD4, on the outer surface of this white blood cell and the GP120 of HIV, that allows the HIV to recognize, bind with, and eventually fuse with this living host cell. So ultimately, HIV fuses with this T cell here, releasing its capsid and all of its contents into the living host cell. The next thing that occurs is the capsid is broken down, so the capsid is disassembled, releasing the viral RNA as well as those enzymes that we mentioned previously, namely reverse transcriptase and integrase. So once the genome, the RNA, is liberated into the host cell, reverse transcriptase then takes the viral RNA, reads that, and produces a corresponding piece of DNA. So the DNA that's being produced is shown in yellow. Ultimately, we end up with a piece of DNA, double-stranded, that represents all of the viral genes. Next thing that happens is that viral DNA makes its way into the nucleus of this cell. Keep in mind that white blood cells have nuclei, even though red blood cells do not. So the viral DNA makes its way into the nucleus. The enzyme integrase allows that viral DNA to incorporate itself into the chromosomes of the living host cell. So now we have the viral genes embedded within all of the normal genes that are part of this human host cell. Those viral genes can then eventually be expressed, producing new copies of the viral RNA. They can then be translated to produce all of the proteins, all of the capsid proteins, the integrase enzymes, reverse transcriptase, all of the components needed to produce new viral particles. So those viral particles are assembled. There's another important enzyme called protease, which results in the formation of the capsid proteins. So ultimately, what's produced is a new viral particle, complete with capsid proteins, copies of the viral RNA, integrase, reverse transcriptase, everything needed to make a new HIV particle that's ultimately released from the living host cell. So now this HIV is released and it's free to go on and infect other living cells. And unfortunately, what can happen over time is that when HIV infects a white blood cell in a person, it can eventually lead to the death of that particular cell. So what can happen is when a person is infected with HIV, over time there can be a very distinct decline in the T cells, and this is a huge problem. It turns out that HIV itself doesn't directly result in the death of a person infected with HIV, but what happens is HIV causes the T cells to die, so that eventually a person infected with HIV, if they acquire AIDS, they simply become unable to mount immune responses to other infections. And so unfortunately, those people end up having severe problems as a result of secondary infections caused by HIV. So that illustrates just one example of a reproductive cycle of a virus. Let's now wrap this discussion up with something even simpler, even smaller than viruses, that can serve as infectious particles. So there are these things called prions. These are simply infectious proteins. So recall viruses, you have to have some proteins, you have to have some genetic material. Prions are literally just proteins that can cause various diseases, and again, they are infectious. So just a few examples of diseases caused by prions: mad cow disease, Creutzfeldt-Jakob, and also chronic wasting disease. Again, these are diseases caused by prions, which simply are infectious proteins, so even smaller and simpler than viruses. That concludes our brief discussion of viruses.